The talk is really an invitation for all of you to join us in MBON and to maybe form a local chapter or regional chapter that helps us organize across the world and using standards on how we collect data, how we manage data, and then how we share data in a more open way so that we can understand what is going on at large scales and regionally. So there's tremendous economic importance for the ocean, of course. The ocean covers over 70% of the planet, but in terms of the economy and jobs, it already is a pretty large basis for many countries. So the Organization for Economic Cooperation and Development published this report a few years back that estimated the ocean economy, or what many people try to call the blue economy, as in the order of $1.5 to $2.5 trillion US. And now they also project that this is going to increase over the next 10 years to over $3 trillion. And with that, so there's a lot of emerging industry sectors, including marine aquaculture, continuing oil and gas and other types of energy, deep sea bed mining and biotechnology, for example, that also involve a lot of jobs. All of these, in fact, the majority of these require some form of biological information, be it from fisheries or tourism that is attractive because an area may be more or less diverse biologically, but also aquaculture. What are the best species and how do you minimize impact on the regional ecosystems? But also other industries require environmental impact statements that would really benefit from having a more organized way of collecting and publishing biological data. So all of this is really relevant from an economic and jobs point of view. Now, what Dr. Sanjeevan described is that there at present is really no organized, large-scale, even national to global effort to observe life in the sea. Everything that we do is through smaller projects or a monitoring effort that is very localized. The data are collected and stored in specific formats that are unique only to that program or project. And so what we would like to do is to try to develop a community of practice that changes this. So this is a major job that we're not talking about something that is going to be easy, but that it includes major efforts by all of us, is how we change this. So the reality is that in situ observations around the world are very sparse. And you can see that in a graphic like this, which is part of the Ocean Biodiversity Information System that Dr. Sanjeevan mentioned. The OBIS has nodes around the world. They aggregate the data as the contributors desire, and you get the data available on pretty openly and publicly through an open interface on the web. And what you see, for example, if you wanted to compare the ocean records in OBIS to satellite data, you will look at the surface ocean, and you get something like this. This is the data collected and available publicly through OBIS over the entire world. And you see that most of the world is just not covered even once. And so, and that's true, it changes over time as well. If you go back in time, you can see in the upper right-hand corner that the number of records that have been stored in OBIS, and that about 10 years ago, this decreased. And the reason that you had a peak there after 2000 was a program called the Census of Marine Life, which in fact established OBIS. And they made a big effort to try to aggregate data, convince people to share the data. And now we are in a situation where the number of contributions to OBIS has decreased substantially. There's also the issue that the biological data is not real time. We collect it. In many cases, this has to go back to a lab. It has to be processed. It has to be quality controlled. So sometimes months or years go by before a biological data record is quality controlled so that it can be put into OBIS. And yet we're all advocating that we have to have sort of a better way to have the pulse of the planet, especially why and how life in the sea is changing. And if the information is not current, if the information is 10 years old, it's very hard to make decisions on the spot on current changes, just because the information that we're looking at is not updated on a continuing basis. This is a real problem for all of us. So the conversation is, why do we have to have biological data? How can we collect it? How can we move it to an assessment tool faster? And that's the goal and the vision of the Marine Biodiversity Observation Network. So we're trying to develop the community of practice that develops common methods, standards to accomplish the observations, but also to share them and analyze them in a common way. So I'll tell you a little bit about the social science aspects of this, how we're organizing ourselves around the world to try to accomplish this. And we're using very large intergovernmental bodies to try to convince people, especially governments, that the way to do this is in a collaborative way, both within countries, but also among nations. And there's two major branches of international government organizations that are trying to do this. One, I'm going to talk first about the left-hand side of this graphic, where we have the United Nations and specifically UNESCO. Within UNESCO lives the Intergovernmental Oceanographic Commission, or IOC. And the IOC has a number of programs or projects, including the Global Ocean Observing System. So also within the IOC are several educational and data organization or management bodies, including the Ocean Best Practices System, which is a database of methods, which I encourage you to check out, the Ocean Best Practices System, and OBIS, the Ocean Biodiversity Information System. It changed its name from Biogeographic to Biodiversity. Now, GOOSE, the Global Ocean Observing System, is a consortium of regional alliances around the world that are that effectively are managed by government entities in a regional sense, and they define, or they're trying to define, the requirements for specific ocean observations, and so they call this the essential ocean variables. The essential ocean variables are the specifications are curated by GOOSE. They're posted on a website, and they provide a guidance for people that are either have observing systems or developing new observing systems for what variables to measure, and hopefully to use common methods to try to then make the observations comparable between regions, but also over time. On the other side, on the right-hand side, we have the group on Earth observations, and it turns out that it's the same countries that are involved in UNESCO that have maritime and terrestrial observing systems or agencies that, for example, the weather agencies or ocean observing agencies or fisheries agencies. So the group on Earth observations focuses on how to organize these agencies in different countries to better coordinate data about the Earth and Earth ecosystems. And so within that, there's something called the Group on Earth Observations Biodiversity Observation Network, GeoBON, which focuses on ecosystems and biodiversity. And there's a marine theme within GeoBON that we call MBON. So on that side, GeoBON has defined a set of essential biodiversity variables. So, and I'll talk a little bit more about these essential variables. So you see that there's a kind of a dichotomy between the essential ocean variables on the one side, where you have a number of agencies talking about EOVs, and on the other side, you have another group of people talking about essential biodiversity variables. So with MBON, we became quite concerned that these conversations were happening in parallel and not in a way where you could have convergence. And we need to have convergence to have some type of hope that we can manage ecosystems using information. So the whole point is to generate information that is useful to people, and down at the bottom, in the bottom box, you have a number of national and international organizations that require this information, either to develop indicators, to understand change, and then make a decision on how you manage that change or manage people so that we can mitigate change or make things better. So this is these diagrams are what the two sides, on the left-hand side, GOOSE, and on the right-hand side, GeoBond, developed for these essential variables. So the essential variables concept was developed to try to understand climate change. And so it first came up as a way to monitor climate. And so they developed the essential climate variables, including air temperature, humidity, that sort of thing. But people realized that this concept of having specific measurements collected in specific ways and sharing the data openly is essential for us to understand weather and climate. And so GOOSE and GeoBond developed the essential ocean variables and essential biodiversity variables with the intent of understanding the ocean and for understanding biology and biodiversity in general. So the intersection in the red area in between the different circles here, that Venn diagram intersection, is what we call the essential ocean variables for biology. And they go as the most fundamental level of observations that you have to measure the essential biodiversity variables. So the essential ocean variables, and I'll go over them in a minute, are the basic measurements of biological, so phytoplankton biomass and diversity, or zooplankton fish biomass and diversity. They go into the essential biodiversity variable framework to aggregate maps. So essential biodiversity variables, if you can think of them, are maps that change over time. So you have to have a space component that in the ocean can be three-dimensional, but it also changes over time. And so you can do that for genetic composition, for species traits, so length of fish or color or phenology. You can also have community composition, ecosystem structure, and ecosystem function. Those are the dimensions of biodiversity. But you have to have the essential data to build those. And so that's the linkage between these two frameworks. Now, you saw the map of ocean observations of biodiversity around the world, and it's pretty poor. Now, what we would like to do is work jointly with the Global Ocean Observing System, which has developed over the past 30 plus years as a system to collect physical data, so temperature, salinity, some of the chemical tracers. Also, so there's a biogeochemistry component, but we would like to layer biology on top of that. So to have GOOSE be a multidisciplinary system for observing the ocean. And so these are the regional alliances that work together in a council to try to standardize how you do this. Some of these are more active than others. You can imagine that in some of the developed nations, these regional alliances are quite active and collect a lot of information and are trying to organize the system better than in other places where things have not evolved so far. So one of our jobs is to try to stimulate the collaboration and development of these regional alliances. These are the essential ocean variables, and I'm not going to go through this. You can go on the GOOSE website and find these, but they're typically there are three groups that are trying to define these, and one focuses on physics, so everything from ocean state to salinity and temperature. Biogeochemistry that deals with nutrients and things like the carbon system that affects ocean acidification. And then you have the biology and ecosystems panel on the right-hand side. And the truth is that these things are not independent. You have to have environmental measurements. So the two columns on the left complement the biological observation. You cannot understand one or the other without them. So the question is, what do you measure given limited resources and time? And so that's the question that may have a local a local design as you measure some of the physics, some of the biochemistry, and the relevant biology and ecosystems in a way that is standardized so that you can then compare the data over time and with different locations. So this is a different way to look at the essential ocean variables for biology and ecosystems. We divided these in terms of functional groups. So for example, you have microbes, plankton, invertebrates on the bottom, fish and turtle birds and mammals. But you also have habitat state, including, for example, shallow tropical coral reefs. And there's big concern about coral cover. The same thing with macroalgae, seagrasses and mangroves. And so each one of these is an EOV. And as you can imagine, these are also related with each other. Fish live in seagrass and mangrove habitats and so on. So there's strong connections between these that we need to be fully conscious. Now, this is another diagram that tries to help understand the relationship between these different groups. So on the upper left, we have goose with, for example, the Indian Ocean Regional Alliance. In the US, we have something called the Integrated Ocean Observing System, IUS, and there are several of these around the world. They define the requirements for operational ocean observing. Operational meaning that it has to be sustained, collected all the time in standardized fashion so that it can inform decision-making. Now, they define the requirements and define the essential ocean variables. The MBON, working also with other groups, for example, the Animal Telemetry Network and other groups, are defining the community. How do we convince people to use standards and common ways to measure parameters so that then we can understand the habitat that we're trying to manage? And we generate the products through groups like GOOSE and OBIS. So the data that are collected by the people measuring biological variables is passed on to OBIS, and products are generated by the various organizations that are like the World Conservation Monitoring Center, for example, of UNEP, or GOOSE itself, or OBIS. So this then comes back, has a feedback loop that if you generate a product and users have another requirement, this requirement gets passed on to GOOSE. They update the essential ocean variable definition, and that gets passed on through this entire loop. So this is what is called the framework for ocean observing that the GOOSE has developed, and developed this about almost 8 years ago. Now, this is another way to look at the process. The GOOSE listens to international organizations, conventions, national governments for requirements. This goes into the process that is, this is typically called DPSR, the Drivers, Pressures, State Impact and Response Framework. And the requirements are passed to observing programs like yours in the Indian Ocean and in the US IUS, but there's many other ones that we're trying to connect. So the question is, can we improve the way that we're doing things now? Can we fill in gaps? Can we work better together? Can we share data, which is a very sensitive issue for many people in many countries? Can we lower the cost? If we want to increase the number of observations, we have to lower the cost so we can deploy sensors in different places. Can we make the data available more rapidly so that addresses data latency? And shorten the time that we at present have where we need the information to make decisions, and having a way to organize data in a way that we can share tools to look at tools. So the strategies that we have under MBON is to understand the metrics of biodiversity and use common ways to aggregate the data in a synthesis way so that we can make regional maps like the Seascapes that we have on the right-hand side. Again, we have on the left-hand side a number of groups that are maybe connected or largely disconnected that collect data. We want to aggregate the data into different tools that synthesize, so the data doesn't have to all live in one place, but it has to be in a way that is compatible, that machines can find them and it can be aggregated using different tools. So infographics, for example, are a simple tool where we can have representations of ecosystems that are easy for a decision maker to understand. And you click on a symbol and you can get a time series that it allows you to understand change and maybe the driver of the change. So this supports, for example, the targets and indicators of Sustainable Development Goal 14, life below water, or other groups. For example, in the US, we have the National Marine Sanctuaries, but there are many marine protected areas around the world that require similar information. So this is our portal, marinebond.org is a way to access the, so it's marinebond.org. I'm going to talk a little bit about the different technologies that we have available for measuring life in the sea. And I'm going to talk a little bit about examples. So these are just examples of technologies that we have to look at microbes and phytoplankton, zooplankton, fish, predators in the ocean, and the bottom. This includes everything from satellites to acoustics technologies, omics technologies, and different platforms with samplers. But you can see that we're starting to populate matrices like these, where the technology is becoming available. It's still not very affordable, but it's becoming available. Now, India has a tremendous infrastructure for data management in information technologies, but also very large industries in electromechanics and so on. So it would be very interesting for India to take some of these technologies and make them cheaper. Here's one example of a device that is now sold commercially, and it's called the Imaging Flow Cytobot, the IFCB. And this is made in Massachusetts by a company called McLean. And what it does is it counts cells. So it's at the size level of phytoplankton. It also measures the fluorescence of these and tries to automate them through artificial intelligence, both by the color, by the shape of what you see in the photographs. And then you can develop time series. This thing can be anchored off of a pier or put on a buoy. And now they're being put on ships as part of a flow-through system, or you can lower them as part of a rosette on a CTD. So the point is to get continuous measurements of very small organisms and understand the composition of the biological community. So this is an example of results of a paper from a group led by Heidi Sosik at the Woods Hole Oceanographic Institution. You also have something that looks at larger particles. So basically looking at zooplankton size organisms. And this is a video camera that you can put again on a CTD. You can put on they're trying to put this on buoys that, for example, the Argo floats that go up and down in the ocean. So if you can miniaturize these devices and have images collected that can then be transmitted to an artificial intelligence engine, you can classify a whole range of particles from phytoplankton to zooplankton. So this thing is called the Underwater Vision Profiler, and there are other optical devices to look at zooplankton. The Continuous Plankton Recorder is one of the oldest devices that is deployed in the sea behind a ship. They're deployed behind ships of opportunity, and it functions by taking water in through the nose, and it filters the water onto a strip. This strip then holds a record of the zooplankton and phytoplankton over hundreds of kilometers over which the ship traversed. Now, if you look at the time series, they're very localized. There are a lot of in the North Atlantic, some in the North Pacific, a few in the South Pacific. I understand there are some in the Indian Ocean, but the time series are also very far and few in between. So this is an important technology that is good, is reliable, and it provides long time series. An area that is really becoming available to science and not just to fisheries is acoustics. So there's active acoustics and passive acoustics. With active acoustics, you emit a sound signal and you listen to the return. And with that, they're looking at different frequencies and the strength of the return. You can look at a whole range of larger organisms, from fish all the way to whales. So you can look at birds, you can look at zooplankton. You can do that in combination when you're looking at ocean physics with acoustic Doppler current profilers. There's information there that we have not fully used to understand biology in the sea and how it's changing. Animal telemetry is very big, very exciting, and it's not just tracking the animals, but the animals now can measure ocean variables. So you can put sensors on animals to measure temperature, salinity, depth, light, the physiology of organisms, and then do that along the path of where these organisms go to feed or migrate. So very, very important technologies. And now there's a lot of groups collecting animal telemetry. It's an issue to have people share these data. But what we need is an open system where these data are shared to understand how different communities of organisms are migrating, why, when, where, and to help this, and to use this information to help manage fisheries, including bycatch. One of the important technologies that is emerging very, very rapidly is environmental DNA. This is part of the whole omics family of technologies, in which you take a water sample and you process it to understand what type of organisms are in it by looking at different primers in the DNA. So for example, with using these different primers listed on the right, you can look at different groups of organisms, be it either the microbes with 16S rDNA or phytoplankton and other things using 18S, or the CO1 primers for a whole range of organisms, or looking at larger organisms like vertebrates using 12S DNA, all by grabbing a sample of water and filtering it and processing it. So this is becoming very important for people interested in quick and rapid surveys of biodiversity. So who lives out there? And there's efforts to try to understand if we can take that information for abundance measurements. This is an example. You can detect anchovies. You can specifically say where and when birds, different types of birds were in place, or different marine mammals. Some of this technology, specifically something called the environmental sample processor, is now being deployed on automated systems like this underwater vehicle called the long range AUV, being constructed by a group called the Monterey Bay Aquarium Research Institute in California. And it's also, some of the technologies are being deployed on something called the Biogeochemical Argo. If you look at the right lower graphic, all of those little points are biogeochemical Argo floats that include these measurements, some of which are important for biology. So you have oxygen, nutrients, pH, chlorophyll fluorescence, and some additional information like zooplankton images that can help us really get to biodiversity. So this is where things are going, and combined with satellite data, we're starting to get a little bit of filling the gaps in our global maps. All of the data, by the way, collected by Biogeochemical Argo and by satellites is freely available. And this is a major change in the culture of our community, is sharing the data openly. Another thing that we have using satellite data, in this case specifically merging ocean color with ocean temperature and some ocean physics parameters, is trying to understand the biogeography of the ocean by looking at what we call seascapes. So we classify the data using thematic classifiers, so basically a clustering system where you end up identifying regions of the ocean that have similar characteristics at different times and in different places. So with that, and what we would like to do is invite people to use the seascapes, which are again freely available using the NOAA Coast Watch website at the bottom, to understand if these data sets can help you predict or nowcast the type of organisms that you have in your region at any one time. An example is here. This is southern Florida in the United States, where we are using the seascapes and in situ data collected by ship to understand whether the seascapes are a good tool to understand the distribution of phytoplankton species and communities in this region. Because it's just really costly and time consuming to go out with a ship. So if this works, it would be fantastic. And we need to validate this around the world. Another way to use satellite data is to look at patterns. Many of you may already do this. We can automate the collection of data within points or boxes or regions to understand how things change over time and detect change. So a very important driver here is detecting change. So we have time series. We can detect if a coastal plume moves offshore into an area where we're interested in, for example, an offshore marine protected area. So we are trying to inform, I'm not going to read all of this, but we're trying to inform government agencies that develop satellite sensors about the requirements for improved spatial resolution, improved temporal resolution, and also to try to measure biodiversity from space. And for that, you need hyperspectral data. So there's an effort, and this is true even in the region of India, where there you have a very strong space program to develop sensors. So this is an important element of the observing strategy. Again, all of this is to organize data in a way that you can generate products. So the conceptual model is to bring data from different data streams. For example, on the left-hand side, you have a lot of different possible data streams about different habitats, and develop tools that you can generate time series about, where you can easily send out a graphic to a manager. The manager is not going to go through all of these data sets and try to figure out what is changing. Typically, managers need simple tools to access very complicated data. And if you try to combine physics, biogeochemistry, and biology, the data sets are multiple, completely distributed. They're all in different formats in different places and very hard to put together. So part of the effort that we're trying to organize is facilitate that, that information management process. So that is the role of MBON. We're trying to make data engines like this, where we synthesize different data sets into pages where a manager can go and easily understand whether the temperature is going up and down or is an anomaly, and whether there's a concurrent change in biology or in the composition of the biological community at any one location. And this is the infographics example, where you have a graphic that is relatively simple about beaches, or maybe a pelagic environment or a benthic environment, where you have silhouettes of different organisms. And if you click on a specific organism or a specific habitat like seagrasses, you can get both the physical, chemical, and some biological information about how things are changing, maybe get trends and the status. All of this is in preparation for the Ocean Decade, the UN Decade of Ocean Science for Sustainable Development, which just started in January. This is a call for us to work together, working on a joint program to look at life in the sea. So we, for example, made a proposal for a program to bring these concepts to the international community in something called Marine Life 2030. So Marine Life 2030 is not just about Sustainable Development Goal 14. There's 17 goals. Many of them are interdependent, and many of them are specific for addressing issues of equality, poverty, how sustainable development in general. So we would like to work with the international community to organize all of these tools in making products that are useful to advance these goals. So this is the framework. I invite you to participate in either through the regional Indian Ocean Goose or MBON, or both, to engage with us in defining user needs, applying these concepts. I'm not going to read all of this, but basically, I think that the whole point is at this stage is helping us work with you in the region, either by forming regional groups that connect to international programs and advance these ideas. I can share this presentation so you can see the details that are listed in here. In terms of data management, Dr. Sanjeevan said that there's a regional issue with data management, but I can guarantee you that this is not a local problem. This is a very serious international problem that is true even in developing in developed nations, where we can use the regional alliances of GOOS as coordinators to support the enrollment of biological data into these engines like OBIS or MBON, or working together. We need to agree on standards. For example, for biological data, for taxonomy, we use something called Darwin Core, and that's part of OBIS and also the Global Biodiversity Information Facility. We use ERDAP to distribute the data, and that has to be picked up and internalized by many different groups. And so there are different standards for acoustics data, for satellite data, for omics data and eDNA. All of those have to be converged and used by different groups. We need to link our portals. So the physical data at the moment lives in different places than the biological data. Ideally, we want to link those. And we also have, and we know that there are very large amounts of inaccessible data. So there, a data archaeology program that is carried out by nations and agencies to try to rescue that data is very important. The physics community did this in the 1980s. And so now they have important data sets that help us understand the baselines of the ocean up to 100 years ago. So regional and national global products have to be defined, integrated in a way that can be stitched together and understand how the planet is changing in an easier way. And that may be done with easy tools, with open software, and common data views that we can all use. And the technology to do all this is available today.